So I presented this morning another phase of our work on a very important landmark trial, SWOG-8814. And that study looked at women with node-positive and estrogen receptor-positive breast cancer who were treated with either tamoxifen alone, which was the standard when the study was started, versus chemotherapy followed by tamoxifen. And we showed that chemotherapy improved survival for these women. So that was the first stage. The second stage, we took tumors that had been donated by the patients on the trial into a bank and analyzed those tumors for expression of 21 genes, the 21 gene recurrence score, or oncotype. And we previously published that for women whose tumors have a very low recurrence score result, chemotherapy did not show a benefit, whereas for women whose tumors had a high recurrence score, the chemotherapy showed a major benefit. So we used those same samples for the data I presented this morning, and instead we went beyond the 21 genes to 20,000 genes and used next generation sequencing technology called RNA-seq to look at prognosis on tamoxifen alone and prediction of chemotherapy benefit. So we found that for prognosis, for the group on tamoxifen alone, there were a lot of genes that predicted early relapse and very few genes that predicted relapse beyond 5 years. Now that's when we looked at single genes. The intriguing part of this discovery analysis was using a software called Cytoscape, where we could look at the interrelationships and the networks these various genes were found together. So these are called metagenes. And so these metagenes, we found 5 of them that were important for prognosis and could describe a group that had almost no chance of relapsing, even though they had positive lymph nodes, out to 10 years. We also found a group, similarly, that did have a high chance of relapsing. So we went on then beyond prognosis to look at, could we use these metagenes to predict better who would have chemotherapy benefit or not. Very intriguing data were that the 3 metagene model found a low risk group that did much better with tamoxifen alone and worse with chemotherapy. On the flip side, those that had a high metagene score with the 3 metagenes did better with chemotherapy followed by tamoxifen. So again, we're coming up with a group of patients with positive nodes who have now 20,000 genes analyzed, and we find these various metagenes that tell us who does better with endocrine therapy alone and who does better with chemo followed by endocrine therapy. And we hope to validate these results in our large prospective study called Responder, which has finished accruing thousands of women, and now we're following these women for late events, and we bank their specimens so we can validate this work and that study going forward.